Welcome to today's webinar on unlocking microbial genetics, leveraging phenotyping for comprehensive analysis. We will dive more into cellular phenotypic insights with Biolog Lab Services. My name is Sandy Almasu, your moderator for today. And our star for today is Cherie Garrett. She's our field application scientist based in Chicago. She received her PhD in microbiology and immunology from University of Illinois, Chicago, but she works now with a lot of our Biolog customers and end users to optimize their cellular phenotyping and identification projects. Cherie, thanks for being here with us. One quick reminder, if you got questions or thoughts, please drop them in the chat. We'll dive into the Q&A with Cherie at the end. So today's plan is very simple. Cherie will give us insights about cellular phenotypic testing using our Biolog Odin. Then we will end the webinar with a quick history of Biolog and our other services, then followed by your chance to grill her with questions. We're looking at about 30 minutes of awesome content. So I'm going to pass the mic to Cherie. Cherie.
Thank you, Sandy, for the introduction. I'm excited to be here with you guys today to discuss how you can confirm your genetic outcomes with our microbial phenotypic microarray services. So this technology focuses on understanding cellular metabolism and using that understanding to characterize cellular phenotypes. The core technology can be used for many different applications, but today I will focus on how phenotypic profiles can be used to understand functional changes in microbial strains and microbial communities. So to give you a microscopic or a macroscopic example of the importance of understanding cellular phenotypes, I wanted to present you with this image of hydrangeas. So as you can see, the flower color of hydrangeas can vary significantly from different shades of pinks and purples and blues. And you would think these different color varieties have something to do with their genetics. But in fact, they're all producing the same color pigment. However, the color presentation depends on the conditions of the soil in which they are propagated in. So in acidic conditions, the hydrangea tends to produce flowers that are blue or purple in color. And this is due to aluminum that is present in the soil, which is more readily available in acidic conditions. If the soil is more alkali, this limits the availability of these aluminum ions, and the lack of aluminum will change the biochemical pathway that produces these pigments and will produce flowers that are more pink or red. So other than this being a fun, cool pH test for soil conditions, we're unaware of any ecological or evolutionary reasons as to why hydrangeas produce these different color flowers. But if you're like a supplier of hydrangeas and you're trying to market to a specific color, it's worthwhile to know or understand how to modulate these soil conditions to produce your desired result. But in more cases than none, phenotypic changes that respond to an environment are crucial for an organism's survival. Our technology requires measuring what a microorganism can eat and how efficiently it can turn that food source into fuel, and whether that fuel is used for respiration or growth. So these food sources are chemical substrates that are usually macromolecules, so carbon, nitrogens, phosphorus, sulfur compounds. And breaking these molecules down is controlled by enzymes and other cofactors like NADPH. So for our chemistry, we use a redox respiration reporter dye that is colorless when oxidized, and when it gets reduced during these metabolic processes, it turns purple. And we measure this purple color to answer a few questions, such as what are the microbes eating? So by putting them in very minimal media and providing them with one substrate at a time, we can determine what substrates that they prefer. We can determine how much they like to eat this particular substrate by quantitatively taking measurements of that dye reduction. And we can determine how fast that they like to consume these substrates by taking kinetic measurements over time. To show an example of a research study that used Biolog technology to learn something novel about a microbe, I want to present to you the wheat plant. So this is an image of a wheat plant that's infected with a fungal disease called Fusarium. This fungus produces a mycotoxin called trichothecenes, which is a major disease of wheat crops and is responsible for major losses of grain worldwide. So high concentrations of this mycotoxin can contaminate the food supply, causing a wide range of toxic pathologies. So it's important to know that the toxin produced by Fusarium really depends on the environment in which they're grown in. And so this group out of Australia genetically engineered a strain of Fusarium where the promoter for the trichothecene biosynthesis gene was fused with GFP. They then use a series of R plates that makes it really easy and fast for them to screen through a plethora of different conditions to identify metabolites that induce production of this mycotoxin. They used one of our plates that contains about 95 different nitrogen sources, and they found very strikingly that the amount of toxin produced is dependent on a variety of amines, specifically amines such as arginine, putrescine, guanine, and agmatine. They also saw that the level of toxin expressed after induction by agmatine was very similar to an actively infected wheat crop. So this is a really nice example of how fast it is with biolog plates and instrumentation to be able to screen through many different conditions at once and quickly find conditions that lead to something that you're looking for, like in this example, increased toxin production. And of course, understanding these conditions or even the biosynthesis that leads to the increased production of this toxin is really important for finding better alternatives for better food storage or fertilizer. So this is just one example of many that is available out in the literature. So the link between a genotype and a phenotype is very complex. And we've known for a while that the expression of a genotype or phenotype is not a one-to-one correlation. In fact, multiple genes can influence a single phenotype, or a single gene can influence multiple phenotypes. Therefore, phenotypic approaches can offer a more comprehensive or holistic view than just a genetic one. And phenotypic information can tell you more about the functional traits of an organism or a cell. And understanding how microbes behave, either when presented with specific metabolites or competing with sources with other microbes, is much more valuable than just sequencing them. So this brings me to Odin, which is our all-in-one solution for cellular characterization. It's capable of phenotyping a variety of microorganisms like bacteria, yeast, and fungus. What it does is that it can incubate and measure any of our phenotyping microplates at multiple wavelengths, allowing you to measure both dye-based metabolic reduction as well as classic turbidity measurements to assess cellular growth, ultimately providing you with robust, actionable data to allow you to hone in on specific conditions that impact growth or cellular respiration. Not only that, but you can analyze side-by-side phenotypic characterizations of your organisms to fine-tune minute differences between reference and test strains. And also, we use the same phenotypic profiles to generate microbial identifications of almost 3,000 unknown microorganisms. We have a series of plates called phenotype microarrays that allow you to quickly and efficiently test thousands of conditions to get a phenomic analysis of your cells of interest. So you can see here that we have a comprehensive set of 20 microplates with pre-filled substrates and inhibitors that allow you to test for the presence or absence of a known biochemical pathway. All of our compounds are known intermediates of cellular metabolism and can quickly tell you a lot about your organism and what it's capable of doing. The first few plates are focused on carbon, nitrogen, and phosphorus and sulfur compounds. And these are mostly our metabolic panels assessing substrate utilization. We also have 2 plates assessing pH, ionic, and osmotic conditions. And finally, we have our last set of plates, which are our chemical sensitivity assays. Specifically, we have a chemical inhibitor that is titrated down into 4 different concentrations. So this looks like a large set of plates, but that's really where Odin comes in, and it allows us to generate these phenotypic profiles in a high throughput manner. So to get into a more biochemical view, you can see in this simple diagram, we're showing the 3 major pathways for energy production. So in the center, we have the electron transport chain, which is flanked by the TCA cycle and the glycolytic pathway, which both feed electrons into the electron transport chain, making NADH from NAD. So for example, one of our many tests has glutamine as an energy source. So if the cell can metabolize this glutamine, it could be converted into alpha-ketoglutarate, and then we have a series of reactions will occur, resulting in the addition of electrons to NAD to make NADH. And these electrons subsequently get passed through the electron transport chain, and the redox side will pick up these electrons and generate that purple color. On the other hand, you can feed a substrate like the sugar fructose, which gets metabolized through a different pathway. If the cell can utilize it, it'll enter the cell, it gets phosphorylated and gets converted into glucose 6-phosphate and goes through the glycolytic pathway, where again, NAD gets converted into NADH, feeding electrons to the electron transport chain and generating that functional metabolic purple readout. So as you can see, we're essentially feeding the cells many different substrates where we're testing the metabolic circuitry that's going on within that cell. So using Odin and our phenotype microwave plates, you can simultaneously screen through thousands of culture conditions at the same time and generate valuable kinetic plots. So here's an example of a typical experiment comparing two cell types. So we have a wild type and a mutant, and we're looking at the utilization of two substrates. So we have substrate A and we have substrate B. On the left, the two strains are able to metabolize substrate A equally well, while on the right, the mutant strain shows a much shorter lag phase and clearly has a different shape to its kinetic curve. So if you only looked at an endpoint measurement, say after 24 hours, you would miss some of the extreme differences in their behavior and assume that they metabolize substrate B the same way. And so these types of differences can have big implications if you're trying to do something like model growth of an engineered strain. Here's a real-life example that can be found in one of our many application notes. Here we're comparing a wild-type strain of E. coli and E. coli with a mutation in the MALF gene, which also carries a tetracycline resistance cassette. So here you can see that after analyzing nearly 2,000 different culture conditions in a single experiment, which is each of these gray points along this concordance line, we can easily point out significant differences in substrate utilization between each strain. So here's an example of what the software can do. It can overlay the kinetic curves of the two strains we're comparing. In this instance, the blue is the kinetic curve for the wild type, while green is the kinetic curve for the mutant. So specifically, the blue points that are to the right of that concordance line indicate that our E. coli has a loss of function in maltose and multitriose metabolism. If we also look at the points to the left of that coordinates line, those yellow points, it indicates that our mutant has gained resistance to tetracycline, which are those yellow curves that you see. So not only does this confirm our genetic construct, but it also reveals novel phenotypes about the mutant strain, and that it also has increased resistance to sulfamethazenes. So now that we've discussed how important it is to get a phenotype of your cells, I want to talk about what else you can do with Odin, which is measuring growth and respiration at the same time. So in this example, you can see that we're measuring one strain of E. coli. However, we've prepared our phenotype microwave plate in two different ways. One, to assess cellular metabolism, we added the redox dye and measured its metabolic response to different substrates on the plate. And you can see those curves in yellow. We can also assess growth or proliferation by omitting the redox dye and running the same PM plate and measuring the turbidity. And those curves are in green. For example, and so you want to know, why is this important? So for example, there are many different cell phases, specifically in fungus, that you can see that can have different responses to metabolites, even though their proliferation might remain the same. And the inverse is also true, where a specific chemical inhibitor can impact growth, but metabolism and respiration might stay the same, indicating that maybe that organism may prefer food sources for cell wall biosynthesis, but they still may respire and stay alive. And as you can see with this particular organism, it can utilize many different carbon sources that may favor growth or respiration. And these measurements that we can make with Odin shows that just measuring just cellular respiration and just growth really misses the bigger picture of what your organism is capable of doing. Here's an example where you can really see the value in measuring both growth and metabolic kinetics. So here, our wild type and mutant pathogenic strain are inoculated into the same PM plate, and we're measuring their kinetics in the same substrate. However, we're looking at their metabolism, which is measuring the dye reduction on the left, and on the right, we're looking at growth. First, you can see that both strains can metabolize the substrate at the same rate. But if you look at proliferation, the wild type strain has more growth in the presence of the same substrate. And so this data is incredibly powerful, as it demonstrates that despite the mutant having less growth compared to the wild type, it still has a similar metabolic profile, which may be of importance if you're assessing certain components during product development. So here you can see that we're running a single phenotype microwave plate. This is phenotype microwave plate 11, which has inhibitors in a titrated series of 4 increasing concentrations. So on the top, you see the plate without any dye. So we're looking at growth. And on the bottom, we're looking at metabolism that has the dye added. If you look at the well center box, we're looking at the effects of enoxacin, which is a DNA topoisomerase. The higher concentrations of the drug on the far right show that there's no difference between the wild type or pathogenic E. coli strains, and we can see this in both the assay for growth and the assay for metabolism. The lower concentrations of the metabolic plate, we can see activity, but there's really no difference between the two, and that's the plate on the bottom. However, when we run without dye, looking at growth, that top plate, we can see that the pathogenic strain isn't really growing at any concentration, which are those green peaks. And we can infer that it's likely using its energy to stay alive and not replicate. However, at lower concentrations, this wild type strain is able to grow. So this is a really interesting difference that you can really tease apart by doing these types of experiments with Odin. So when you measure both growth and restoration in one experiment, what does Odin tell you? And again, it comes back to what the cells are eating. So if you're feeding them something that is suboptimal, they may be in stress conditions and won't behave the same in an environment that isn't deprived of nutrients. You don't also have to run pure cultures. We also have applications for whole communities, and you can get phenotypic profiles for the whole community, and you can really tell who outcompetes who. And you can also look in, like, for example, if you want to look and see if bacterial respiration is competing with fungal biomass. And if you're trying to make a product with fermentation, you might care about whether it's respiring more than it's growing. So for example, for making beer, you may want your yeast to not increase in biomass. But if you're making a high protein food, you may want a more biomass rich fermentation product. So not only do we have microbial phenotypic characterization, but we also have 3 additional types of analysis. So one solution we have for understanding community level analysis is the EcoPlate. It provides a sensitive and reliable phenotypic method for measuring environmental change in microbial populations. These plates measure the spatial and temporal changes in metabolism in ecological microbial communities, such as soil, water, compost, industrial waste, just to name a few. We can then provide high-resolution kinetic data that reflects the changes in community properties and dynamics that can be further used for multivariate analysis. We also provide phenotypic characterization for yeast mitochondria. As you know, yeast are important in the biotech and fermentation industries due to its use as a human model, their ease in genetic manipulation, and its importance in beer and wine industries. So if you know the mitochondrial metabolism of your yeast strain, this gives you an additional resolution in their function, so you know how to manipulate them if you need to produce a particular product. And because all of this is energy driven, we can provide you with a live cell metabolism and chemical sensitivities and use this to identify any functional changes in your yeast cells or clones. And lastly, using phenotypic profiling to generate metabolic fingerprints for identification. And so using our Gen 3 microplates that contain 95 different substrates and our respiration redox reporter dye, each organism will develop a unique metabolic fingerprint on each microplate. But because we have the incorporation of 90 different tests, we have a greater resolving power, which allows us to accurately identify over 3,000 aerobic organisms, and some even down to the strain level. Now that I've described to you the importance of phenotyping and the solutions that Biolog provides for robust phenotypic characterization and identification, I will now discuss how our servicing platform works and the types of analysis you can expect to receive. Biolog Lab Services team has a large menu of services that we offer, and Sandy will discuss these at the end, but for now, I'm going to focus on our microbial phenotyping with Odin. For a basic overview of phenotypic characterization services, we always provide an initial consultation. This consultation will include a discussion with one of our scientists and sales team members to thoroughly go over the aims and expectations of your project, and we'll create an experimental strategy with a budget and expected timeline to completion. Once agreed upon, and the sample was shipped to Biolog Lab Services, where we will screen through the organism for potential oxotrophies, and this is so that we can optimize the experimental conditions prior to running your requested assay. Well, let's run the organisms on your desired screening microarray plates on Odin, which will collect the kinetic growth or metabolic data over a specified amount of time. We will then prepare your kinetic results by analyzing and interpreting your data based on your project aims. And during this, we may find some parameters or culture conditions that may need to be adjusted, and we will always update the assay as needed. Finally, once the analysis is complete, we will send you the final report, which is followed by a post-project consultation. And here you will have a one-on-one meeting with our scientists to discuss the findings and potentially discuss any additional analysis to be performed or any additional assays you may want to run. In the final report, you will receive comprehensive phenotypic profiles depicting growth or metabolic curves for every condition tested. Here you can see that we're looking at a single phenotype microarray plate, and that it's showing the space-filling overlays of 2 replicates of a wild-type strain and 2 replicates of a mutant strain. And you can see here that you can easily visualize the differences in metabolism or growth between each of the strains. In terms of data analysis, there are many different ways that the data could be analyzed. But here's a comprehensive list of the parameters that can be calculated from your kinetic analysis. We can assess the average height or average Y value of the ODs over the course of the assay. We can assess the area under the curve, which is just the sum of those kinetic data over the time of the experiment. Also the maximum height or maximum Y value, which is the 90th percentile of the kinetic data. But we can also assess rates. So like the maximum or initial rate, which assesses the slope of that best fit line of the data, which are both great tools in fine-tuning metabolic consumption between organisms or communities. And the type of analysis requested during the initial consultation or can be updated during the post-consultation call. Of course, we can provide pairwise comparisons. So if you're interested in comparing, say, a reference wild-type strain and a test mutant strain, you can select any of the aforementioned parameters to quantify and compare the data between your 2 strains. Not only will we provide graphical overlays between each of your samples, but we will also provide a full list of substrates or inhibitors that signify a significant phenotypic difference between the 2 organisms of interest. And these differences may either impact growth or cellular respiration. And this data will reflect which plate was utilized, the well, the chemical, the difference between the 2 strains depending on the parameters selected, as well as a description of the properties of each substrate or inhibitor in each well. So here's an example of the final report you will receive. Each report will have detailed explanation of your samples, including the methods and a high-level overview of your data. Each report will have the kinetic curves for each compound tested, as well as if pairwise comparisons or metabolic or sensitivity changes between reference and test samples were requested. So now we do realize that analysis through Biolog Lab Services may not be enough, and you may want to do additional analysis in your own statistical software programs. So we'll also provide the raw data in CSV or JSON formats upon your request. So in terms of samples that we can work with a variety of samples depending on your needs and workflow. So for PM and ID services, we can use any BSL 1 or 2 organism, and this is usually provided in an agar, some type of agar or broth culture. And for community level analysis using our eco plates, we can analyze almost any type of sample from soil, sludge, water, sediment, etc. And all of this can be determined and further discussed during your pre-project consultation. So I hope I've demonstrated to you the value of Biolog Lab Services for phenotypic analysis. Our comprehensive consultation and optimization process guarantees optimal experimental outcomes, assay conditions, and data analysis for you and your strains. And our approach utilizes our scientific expertise and our commitment to you to ensure accurate results and providing valuable insights for your further research. And in summary, phenotype microarrays provide a very rapid method to gain functional cellular insights into growth and metabolism using many different culture conditions. And using Odin, which can efficiently monitor growth and cellular respiration kinetics for a thorough understanding of cellular functionality, as well as because of Odin's automation, we can also do community level analysis and phenotypic identification. And then the scientific expertise of Biolog Services can help you design and generate relevant physiological data to revolutionize your microbial analysis. And of course, you have the option of purchasing Odin yourself for your team and have it customized for your specific workflows. And Biolog Lab services can also be used if you're interested in doing a proof of concept study or demoing an instrument for purchasing support.
Well, thank you, Cherie, so much for such a great insight about our phenotypic services using Odin. As you all have seen earlier, the slide, I will dive into other services that we offer. Here at Biolog, we strive to giving our customers confidence in identifying organisms on top of our phenotypic services. So we do offer multi-omic identification services of multi-organisms such as bacteria, yeast, and fungi. So you can look at different services that we offer, like genotypic using our single DNA sequencing, proteotypic using our Bruker MALDI-TOF, and phenotypic identification using our Biolog Odin. We also offer other services such as enumeration, total plate counts, bioburden enumeration, visual ID, gram stains, biological indicator testing, water sample testing, purities, and any special microbiology projects that you're working on and you need assistance in developing a method or any other projects, please reach out to our lab and we can assist you with any other type of services. Next slide. So whether you're concerned about contamination or enumeration, our polyphasic, genotypic, proteotypic, and phenotypic testing services can help you reduce costs, guarantee brand safety. So at Biolog Lab Services, we offer results under our ISO 17025 2017 accreditation. And we also registered with the FDA, GDUFA, and also CGMP compliant. So as we come to the end of our journey, let me introduce you to our ecosystem here at Biolog. We're on a mission to shed light on intricate world of microbes and cells. Through cutting-edge technologies such as Odin and pioneering analysis, we offer fit-for-purpose instruments, a comprehensive database, custom plates, and also accredited multi-omic lab services as we've seen earlier. So think of us as your partners propelling your scientific pursuit forward. Thank you all for joining us on this enlightening exploration. Always remember, Biolog's for you. So on the next slide, you will see actually our contact information where you can reach our Biolog instrument site at info@biolog.com, but you can also reach our lab services team at labservices@biolog.com. So now we'll dive into our Q&A session. We have a couple questions in the chat. So Cherie, do you have other samples of customer use cases and publications?
Yes, so you can find all of that information on our website. We have a very extensive publication database that can last a look has over 5,000 publications. So if you go to our website, you can definitely find all of those resources. You can even filter for certain applications or organisms that you're most interested in and find those specific examples for you.
Great, thank you, Cherie. Another question in the chat it says, I'm curious to know, is there any options for phenotyping the methane oxidizing bacteria? I'm working on aerobic methane oxidizing bacteria. I would like to study them for their phenotypes, but I can't find any suitable Biolog plates for methanotrophs. Could you help me in this aspect? Thanks in advance.
Oh, yes, thank you for this question. So we do have many users who do study methanotropes. So we do have some applications on that. So if you reach out to us, I will be happy to send you some resources and some publications that support its use.
Great. Hi, how make metabolic map based on substrate utilization by using Biolog EcoPlate? How can you make metabolic maps based on substrate? Go ahead, sorry.
No, you're okay. So if you're just interested in just knowing what those substrates are, we can provide you with the plate for the EcoPlate or the plate maps for the EcoPlates. The EcoPlates primarily have carbon sources dry down onto them. So, and they're arranged in a way in triplicates. So you can analyze a triplicate sample of one sample or just 3 separate samples at the same time. So we can provide you those plate maps for the EcoPlates.
Thank you, Sheree. Another question, what is the advantage of this approach, the phenotypic approach, versus just doing metabolomics?
So the advantage of using this approach, I think they go hand in hand. They can both tell you lots of things. I would say that with the phenotypic approaches, is it gives you a live cell characterization. So using metabolics, you might, have to kill the cells or something like that. But with this, we're giving you live cell functional metabolic profiles.
Okay, great. Another question, do you have biolog specifically for virus characterization and animal cell culture?
So yes, we do have plates specific for mammalian cell culture. So we didn't discuss that today because we were just focused on the microbial aspects, but we do have plates specifically for that. So you can look at substrate utilization as well as chemical sensitivity to mammalian cells. A lot of those sensitivity plates have anti-cancer drugs and any other type of chemical that is known to disrupt mammalian cell cellular respiration and growth. So we do have plates specifically for that. For virus characterization, you can use our mammalian plates for doing that type of work as well. And if you're looking at bacteriophages, we do have applications for phages, looking at its capability of lysing those cells.
Okay, great. I think there's a comment also in the chat saying, I would be also interested to know how to use AI to predict some phenotypes based on the data generated in genome.
Yeah, I think that's a great application area. And using AI, we're not familiar with that, but I can maybe find some resources and applications of any folks who've used AI with our phenotype microarray plates to further explore your research interests.
One last question, Sheree. I know that we're over time. Thank you all for staying online with us. We're not in the US. Can we still use your service? So I'll take that answer actually, since it's a more of a lab service question. We are actually able to provide services to outside of the US. So if you have any specific organisms that you're trying to send to us, please make sure you consult with our team. Reach out to info@biolog.com or labservices@biolog.com, and we can walk you through what needs to happen before you send us the sample. One more question before we get off. Would we be able to phenotypically compare lab strains to environmental strains? Example, lab-generated E. coli and environmental E. coli.
Yes, absolutely. As you saw, many of the examples were mostly E. coli, as you saw. So we can definitely do comparisons and looking at the different metabolic profiles between your lab E. coli and your environmental E. coli. We can definitely do that for you.
Perfect. Well, thank you all for joining our webinar. It was great having you all and great questions. And if you have any questions that we haven't answered, please reach out to our team. We'll be more than happy to assist you. Thank you all. Have a great day.